So in the last week, we have discussed on the genetic mutation. We know that the genetic mutation can categorize in two levels, in DNA level, in gene level. So we have, in this level, we have insertion, while a few nucleotides can be inserted into a DNA. A deletion, when a series of nucleotides can be deleted out the genes or several genes. Duplication, while a set of DNA, a set of nucleotide can be replied due to error in DNA replication. We have inversion and we have substitution, while several or one nucleotide, sometimes only one nucleotide, will be replaced by another nucleotide. So in the higher level of mutation, we have the chromosome mutation. We have two types of chromosome mutation. We have the structural chromosomal mutation. We have deletion, duplication, inversion, insertion, and translocation, just the same with the DNA mutation. And we have the chromosomal number mutation, which dealing with the number of chromosomes. So we have two subcategories in this type. We have aneuploidy, where the number of chromosome added or deletion is just one or a few chromosomes. And we have polyploidy, when we have an extra set, the whole extra set of chromosomes. So let's watch a small clip recap about the type of genetic mutation. Okay. So wait for me a minute. I'm sharing the sound.
The term mutation brings countless fantasies to our mind. Right from the ability to fly high in the sky to that of diving deep into the oceans. We all love to have such fantastic abilities. But these are all fictitious. Such fantasies don't exist. So can we say that mutation also doesn't exist? Hold on, that's not true. Mutation does exist. But mutation doesn't lead to such drastic changes in us humans. We can't have mutations that give us extraordinary powers. Rather, most mutations give rise to concern. They result in undesirable changes from time to time. In these upcoming videos, we shall majorly talk about mutation, what causes them, and their types in general. Let's begin with the definition first. Mutation, in simple words, is defined as sudden and abrupt changes to the genetic material of organisms. Now let's break this down to understand it better. Sudden and abrupt changes means a few changes that occur suddenly and are mostly unexpected. These changes are usually random and disruptive to the normal functioning of the cell. And where do these changes occur? They occur in the genetic material of the organism. That means in the DNA. And as a result, either the gene or the complete chromosome gets affected. So mutations are spontaneous changes that affect the DNA or the genes or sometimes the complete chromosomes. Now, since these are the areas that get altered, mutations are categorized on the basis of them. The two major types of mutation are genetic mutation and chromosomal mutation. Let us see these types. Let's consider this small section of the DNA. Let's say that this part changes into another. Another possibility is that either this base gets deleted or an extra gets inserted. In either case, the outcome is that the sequence of the gene will change. Technically, the reading frame will change. The ribosome assembly, including the tRNA, reads the genetic code in triplets. These triplets are called codons. Now, if there's any change in a single nucleotide, then the entire codon will change. Thus, the reading frame will change. And what will this result to? That's right. The altered gene will give rise to a different protein than the usual one. And there are chances that this defective protein turns out to be a harmful one. So any change in the DNA or in the genes can cause a shift in the reading frame. As a result, an altered protein is produced. This was the first type. Now let's zoom out of the DNA structure. What do we see? Yes, we see a typical chromosomal structure. Now tell me, what will happen if there are any alterations in its structure? And what if the total number of chromosomes get altered? Let's find the answer in the next part. This is a representative karyotype of a human. By now we know what a karyotype is. Can you try to give me some conclusions of it? You may say that it looks perfectly normal. But let me spot a flaw for you. Do you notice this set? This is the 21st chromosome set. Instead of having a normal pair of chromosomes, here we find a triplet. Now you may say that it may be good for the individual as it has an extra chromosome. But that's not the case. Any change in the number of chromosomes creates a lot of defects in the normal functioning of the body. On this note, let's understand chromosomal mutations. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? It can happen in two ways. One way is where the structure of the chromosomes gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's have a look at each type. This is the structure of a typical chromosome. Now imagine this small part gets physically removed. This would result the chromosome in getting smaller as its segment is lost. This also means that if the segment had some important genes, then they will also get lost, which can result in severe abnormalities. Such type of a mutation is called deletion. This is where the part of the chromosome gets deleted. Now imagine this part of the chromosome gets duplicated. As a result, the chromosome's length increases. Needless to say, this will also result in some or the other abnormality in the individual. This type of mutation is called duplication. Now comes the next type. These are two different gene segments. Imagine they get flipped. Thus, the chromosome will appear like this. This is called inversion. Do you think this type of mutation is harmful? It may or may not be harmful depending on which genes get flipped. Now let's take one more chromosome. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. What will be the result? That's correct. Both the chromosomes will change drastically. This type of mutation is called as insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interexchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both the chromosomes get altered completely. Any idea what this type will be named as? Well, this type of mutation is called translocation. All these mutations resulting in the disruption of normal chromosomal structures are broadly capped under a single heading. They are categorized as chromosomal aberrations. Now, this was about the mutations that involve changes in the structure of the chromosomes. What about the mutations that involve a change in the number of chromosomes? Let's understand that in the next part. This is a normal chromosome, while this is a disrupted one. When the structure of a chromosome gets altered, It's called chromosomal aberration. We have seen this. Now let's move on to the second type. It's the mutation that involves a change in the number of chromosomes. It's called as aneuploidy. In literal terms, aneuploidy means the presence of an abnormal number of chromosomes in a cell. So the karyotype of a person with trisomy 21 is an example of aneuploidy. The presence of one additional chromosome in the 21st set gives rise to a total of 47 chromosomes in a person. So instead of a normal 46 chromosomes, this person has an additional chromosome. To be precise, an additional autosome. This case of aneuploidy is called trisomy 21. This is because there are 3 chromosomes instead of 2 in the 21st set. The problem of trisomy 21 leads to a condition called Down syndrome. We will get into the details of these disorders in our higher grades. But for now, let's focus on understanding the chromosomal mutations. This aneuploidy involved an autosome. Now tell me, what if the number of sex chromosome alters? So instead of XY, what if an individual has XXY? Is that even possible in the first place? Well, yes. There can be individuals with an additional X chromosome. This condition is called Klinefelter syndrome. Such individuals exhibit both masculine as well as feminine characteristics. Now, in the case of females, imagine that instead of addition, there is a deletion of an X chromosome. This would mean XO. So this individual will have only one X chromosome. This condition is called Turner syndrome. There are a few developmental abnormalities in such scenarios. These individuals have a total of 45 chromosomes only. These were a few examples of aneuploidy that involve autosomes and sex chromosomes. Now, before we conclude, tell me one thing. What causes these mutations? How can DNA or genes or entire chromosomes get altered? There are several factors named mutagens which lead to alterations. Some of them include radiation, chemicals, and even certain consumable items. To summarize, we have 2 major types of mutations. One which involves alterations in the DNA or the genes. These mutations result a shift in the reading frame, resulting in defective protein synthesis. Alterations in the structure results in chromosomal aberrations. They can be categorized as deletion, duplication, inversion, insertion, and translocation. The other is where the number of chromosomes are different than the usual in the organism. It's called aneuploidy. The changes can be in the autosomes as well as the sex chromosomes. Here, the total number of chromosomes in a cell are different from the usual. This was about mutations in general. To find more interesting videos, do not forget to subscribe to our channel.
So next we will study about genetic. Today we'll discuss about genetic mutation, which is leading to the genetic variation in the population. So you all know about Charles Darwin, which is considered as the father of evolution, and he and his famous theory about evolution. When he traveled around the world, he draw like a map of species, which one is close to which species is close to which one, and based on which characteristics. So he set a kind of diagram like this, the cladogram here, and in the cladogram, he has draw a species which is one is close to another and evolve to each other based on a set of clinical features. So for example, in here, we have a salmon, which is a fish species, and the closest one to fish to a salmon is salamander. And the main difference between those is salmon have a salamander have a lung, while salmon do not. And the salamander next step evolved to the lizard, which is have the feature is claw or nails. And when lizards evolve into mammals, is they develop their mammillary gland, which is can feed the offspring and the fur. And when the mammal evolved into chimpanzee, so his main characteristic trait is the position of the thumb in the hand, which is have a possible thumb, which is the position in face of the other fingers. So this is a cladogram. So based on this cladogram, would you help me to draw a cladogram with this 6 fiction species? So in here I have 6 species, A, B, C, D, E, and F. And could you help me to build a small cladogram like this one with these 6 species based on their phenotype feature, please? Just one small question before you start your exercise. Is my internet connection today is okay? Because sometimes I feel it not going really smoothly. When you heard the clip, do you see it play smoothly or no? Okay. Yeah, so okay, I'm asking about my the quality of my internet connection. So I have the answer is A, D, E, C, F, and B. Okay. All right. All right. So all of you have provided the solutions is A number 1, D, E, C, F, and B. And the right answer is yes, it's A, D, E, C, F, and B. So if A row in I, we have D here. If D row in fur inside, we have an E. The E have with this scary teeth in here. We have a C. More fur, we have an F. And if the I turn red, we have a B. So this is an example of cladogram, and evolution follow the Chapman theory. But sometimes to draw a cladogram based on the phenotype is really difficult. So this is just a simple example. So in real life, we will see that sometimes based on the phenotype features, is not always really effective tool to identify the genetic distance, the differences between species and we call the evolution step between species. So for example, in here, we have 6 species of tiger. We have Malayan tiger, we have China tiger, Indochina tiger, Sumatran tiger, Bengal tiger, and Siberian tiger. So each species of tiger have phenotype features a little bit different with each other. So could you help me to do the same with the cladogram you have built very beautifully in the last exercise? Can you help me to build the cladogram with the 6 species of tiger? So with the last exercise, you build it quickly, like in, I don't know, 10 seconds, 15 seconds. Someone have already finished. How about this one? With all these phenotype features. Anyone can help me? Yeah. So we can see here, although when we see at each species, we can see that they are different a little bit based on the size of the eye, the size of the head, but the size of the neck here, like the Bengal tiger, we have a very short neck. But it's not easy to build a cladogram like the previous one with these 6 close species. So as a result, based on the phenotype features, is not always the effective way to build cladogram. So luckily, nowadays, we have these species depend on, so several types of species in here, when they mate with each other, their offspring cannot have the ability to produce offerings. So apparently they are different species. So so luckily nowadays we have a new tool, which is the molecular tool, genetics tool, to identify the differences between those. So DNA sequence is always very clear, and we can identify the mutation. For example, in here, we have 6 species of tropical fish here. And based on the DNA data here, after alignment that we put it in the line to see that corresponding to its position, if we have the similar position in the other species. We can see that there is mutation, and based on the position of the mutation, we can identify what is the genetic distance between one species of the next. Okay, so for example, in here, these 3 species seem to be more close with the other 3, since we have the deletion of the fragment here, the deletion region in here in a common. And this species is considered really close to each other, since they have no difference in the gene of interest. Okay, so let's come to another exercise. So now I have 6 species here, and this is a DNA sequence of a fragment composed of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 nucleotides. So this is similar regions of the genes between the 6 species A, B, C, D, E, and F. So could you help me to build a cladogram with these 6 species based on the DNA sequence of these 6? Would you? So for more convenient for you, I will copy the sequence and put it in the chat box. Okay, so put it in the chat box here. There you go. So very quick, we have a first answer A, D, E, C, F, B. How about the other? Are you finished yet? Okay, we have the second solution, which is the same with the first one. All right. Okay. So we have a few differences in here. Some students have finished. All right. We have a few differences here. D, A, C, B, E, F. Okay, so now I will ask for a student can provide me the solution, how to handle the exercise, okay? And you can share your screen and tell me how you can, based on the DNA sequence here, can tell what is the order of the cladogram in this species. Please, I will invite Vũ Đức Thành, please. Could you help me to handle the exercise? You can share your screen. Okay, so not here. Another one, anyone volunteer to handle the exercise? Anyone can give me the clear explanation about the exercise? You can share your screen to make your explanation more clear with everyone. Anyone? Okay. Okay, so I will provide the solution. So to identify this, first we have to identify the differences between those sequences. So first we will have a matrix between to show the differences between X species. So in here we have 6 species, so we will have a 6 times 6 matrix here, or 5 to 5, whatever. So let's compare the DNA sequence by pair, okay? So first I compare the DNA sequence between the species A and B here. So we can see that in the A and B here, in 12 nucleotide, I have 3 positions are the same between A and B, and the other 9 are difference between A and B. So the differences between A and B is 9. I put 9 in the matrix. Okay. So the next, I will compare between A and C. You see what is the differences between A and C. So in between A and C, I have only two differences between A and C, and the other 10 are the same. So we have the number of differences between A and C is 2. So I put 2 in the table. So compare each sequence with each other, A with B with C, or A with D, A with E, A with F. I will have fulfill the matrix, and I will have the matrix like this. Okay, so this is the number of differences between each pair of species. Okay, so you understand this part, right? So if you have something you do not understand clearly, please let me know, all right? So this is the differences between species. So I will come to which ones you have are most close with each other. Which two species in here are the closest to each other? So in other words, the closest one, that means that between the two species, the number of differences is smallest. And the smallest number in here is 2. 2 is the smallest number. So that means that A and C are the most two species that close to the other, that most close to each other. And also we have 2 in between the B and E. So A and C are 2 species that most close to each other. And also the B and the E are 2 species that the most close to each other. So I put A and C as a group, and B and E as another group. Okay. So for example, with A and C as a group, I have decreased the matrix here, which is the 5 times to 5 triangle matrix, 2 4 to 4 triangle matrix by group A and C as a group. Okay, so now between A and C, I have the distance between AC and B is equal to the distance between A and B and C. So the distance between A and B is 9. The distance between B and C are also 9. So the distance between the new group A/C to the B is 9. Okay. You understand this part, right? Okay. So suppose that you all understand this part, because I heard nothing from you. All right, great. So the next distance, we have the distance between A and D is 4. Distance between A and D is 4, and the distance between C and D is 5. So the distance between the new species A/C with D is equal to 4 plus T. The mean between the two distance is 4 plus 5 divided by 2, which is 4.5. And so on by this method. For example, the distance between A and E is 9. and distance between C and E is also 9. So I have the distance between A/C with E is 9. So based on this method, I have successfully decreased the matrix from 5 times 5 to 4 times 4. Okay. So continue the loop. After I have the smaller one matrix here, so the smallest number in here is 2. 2 is the smallest number. So I continue to decrease the 4 times 4 triangle matrix to 3.3, 3 times 3 by group E, B, and E as a group, because the smallest number here is 2. So the new distance here, so distance between P and slash AC is 9, and the distance between E and slash AC is 9. So the two other here, so 9 is distance between the new group B slash E and A slash C, and so on. Is 3 times 3. So I decrease the dimension, the number of column and row in here from 4 times 4 to 3 times 3. So continue. What is the smallest number in this matrix? So I have here 4.5, exactly. 4.5 is the smallest number. That means that in this new matrix, the smallest distance is this genetic distance between A slash C and the D. So I group A and C and D as a group, because the smallest number in here is 4.5. So yeah. So from this, I continue to decrease the 3 times 3 to 2 times 2 by group A and C and D. As a group, I have a new group A, C slash D. So the new distance between this one and the B and E is 7.5, since the distance in here is 4.5 and 9. So I have the new one in here is 7.5. So the 2 here is keep also the 11. Okay. So now the smallest one again is 7.5. That means I group these two last species here as a group, and the F was in here. So the final solution is A, C, D, B, E, and F. A, D, E, C, F, B. So I check if I have any answer is okay. So anyway, not a lot of people was right. So anyway, do you understand about this, the solution of this part? So nowadays we have a software to launch that to help us to calculate the genetic distance of hundreds of species based on a much more larger DNA fragment. It can calculate with the genome of billions base pair. Of course, with only 12 nucleotides, we can do it manually, but with a longer, like billion base pair, it's impossible to do it manually. We need software to do so. But the algorithm, algorithms to calculate the distance and categorize and draw a cladogram are the same. Okay, so at least now you understand the mathematical calculation behind this. So next part, let's come to the genetic variation. So today we will have a lecture with 3.5 hours. So really torturing. So the genetic distance, genetic variation between people are only 0.1%. That means that the genetics background in all people in this planet, no matter what they are come from, Asian people or African people or Caucasian people, the DNA are 99.9% identical. More than 99.9%, depend on the pair. Only 0.1% of us are different. So when you think that we are differences, please think about this number. We are not really, not much that different. But this 0.1% of our genetic background will decide numerous features that different between us. So we have differences in the physical appearance. Some people was high, some people were short, some people with a different hair color, like black or red hair or blonde hair or brown hair. The color of the eyes, the color of the skin, the freckle in the shape of the nose, the shape of the ear, and so on. We have also difference in a susceptibility or resistance to a certain disease. For example, like type 2 diabetes, or a risk of getting cancer, or cardiovascular disease, so on. And we have also this 0.1% of our genetic variant will decide the different response to a certain medication. So that's why nowadays personalized medicines is really a trendy research, since it's provide a different treatment for one same common disease to different patients based on the differences in the genetic background. So for example, with the same treatment to cancer, like hepatotic cancer, the drug provided normally will be hepatitinib, but some people was so tolerance with this drug. So based on the genetic background, the doctor, the physical doctor can provide the other treatment that more efficiently, efficient for those patients. So based on the size of the human genome, which is around 3 billion base pair, the differences in the of us is around 0.1%. That means we are only different with each other like in 3 million base pair. And for short, nowadays we have known about more than 50 million genetic variation based on the 1000 Genome Project. So genetic distance, genetic variation here can determine different phenotype between from individual to different individual. For example, in here we have some with dark hair, some with brown hair, the shape of the face, the freckle in the skin, the hairline, the amount of hair in the armbar. And sometimes genetic variation can provide some phenotypic features that really extraordinary. So for example, in here, we have met Stephen Crone, as known as the man who cannot catch HIV. So Stephen Crone is a homosexual man, and meanwhile all of his partner, and just for more explanation, the homosexual man is considered that the top risk of getting HIV, based on they always get infected during a relationship. Anyway, meanwhile, all of the partner of Stephen Cron have been killed by the HIV. The man seems to be healthy. Seems that there is no sign that the HIV can infect into his immune response. So when the medical doctor investigates about this case, they find out that in the genetic background of Stephen, they found out that there's a mutation, a delta 33 mutation on the CCR5 genes. So delta, which is short for deletion. So it's a deletion of amino acid 33 on the CCR5 genes. So this gene is encoding for a receptor on the membrane of a T4 cell. So 3. I cannot. Of the delta 33 mutation on the CCR5 receptor. And this receptor was encoding for CD4 plus cell in the membrane of the T4 cell. So when the HIV virus get infected into a T cell, it will inject its RNA inside of the T4 cell. And from this RNA, we have studied in chapter 1, the RNA will be reverse transcribed to DNA. So from this DNA, it to transcribe to RNA again and start to synthesize the protein envelope and replicate into 1,000 more copies of HIV virus. But in case of Stephen Cron, he has a delta 33 mutation. It made that the CCR5 genes encoding for a false structure of CCR5 protein on the membrane of CD4 cell. And this modified compared to the normal one here, make the HIV cannot recognize this is the CD4 cell it had to inject it to transmit its DNA. So as a result, the HIV virus cannot inject into the immune system of Stephen Cron. And in 2018, a Chinese scientist. Actually, he's a physician, and he's in the end of 2018, based on the mutation of delta 33 on the CCR5 genes, have claimed he has created two new baby girls based on modified the genes by the CRISPR-Cas9 tool to protect these two baby girls from infected HIV from the father. So let's hear how he's talk about his research. So I don't know if we go in here. Where is the setting to sharing the sound? How can I sharing sound in here? Các bạn có biết là ở trên cái công cụ Zoom bằng website này thì share cái âm thanh ở đâu nhỉ? Ở trên software thì mình biết, nhưng mà không thấy ở trên cái này, không thấy share cái. Okay. Các bạn có nghe được âm thanh của clip không?
Không.
Oh. Share ở đâu nhỉ? Lúc nãy sau khi bị đuổi ra thì mình không làm cách nào mình vào được Zoom bằng software nữa. Toàn báo là invalid ID meeting number. Không hiểu sao. Ok, các bạn chờ một chút nhé. Okay, so everyone can hear me?
Yes.
Finally. All right. Okay, so who the host now? Anyone who the host? Okay, I'll claim the host. Sorry again.
You may wonder why we
Okay, so I think it's worked now.
You may wonder why we use the gene surgery to prevent HIV. Why not another lethal disease? Two reasons. First, safety. Second, real-world medical value. As a scientist and the father of two
What happened?
girls, safety is my number one concern above all else. The gene we choose, CCR5, is one of the best studied genes. In fact, about 100 million people naturally has genetic variations that disable CCR5, protecting them against HIV. They were healthy. Our surgery caught exactly at the start of that natural variation. Decades of clinical trials has tested medicine to mimic that natural variation, including the first gene editing trial in the United States. Well, it proved safe. Also, protecting against HIV requires the simplest gene surgery, which is to remove just a few DNA letters. Correcting a broken gene will helpful for more diseases like familial cancer or muscular dystrophy. It's more complex. Studies in the US hasn't shown this works well in embryos yet. The second reason we end up choosing HIV as our first target is the real-world medical value. HIV remains a deadly and devastating infection that need medical advances. About 1 million people died of HIV last year. New infections remain 3 times higher than the UN AIDS target for 2020. No vaccine or cure exists. Taking pills daily from life carry healthy risks. In many countries, discrimination deepens the devastation of infection. Employer may fire people after discovering their HIV status. Doctor may refuse to treat. CCR5 surgery, while not applicable to the general public, it may be valuable to help a few affected and very high-risk families protect their children from the same fate. This led to the National Academics in 2017 named vaccine-like disease prevention as an ethical application of gene editing.
So in the lab of Xue Chuanhui, a physician, he claimed that he had created 2 new baby girls. But soon later, many scientists have reported that such genetic modification in human model without any ethical statement could be illegal. And as a consequences, in 2019, Ken Hui have been putting in jail because he had performed genetic modification in human without permission from the government. Until now, because he have never sent his research into any reported paper, so we don't know that he actually did perform the experiment or not. But in the future, with the development of CRISPR-Cas9 and the genome editing tool. And we know that the small and simple mutation and CCR4 receptor can simply block the injection pathway of HIV into the CD4 cell. The reported HIV genetically modification non-organism could be possible. So next come to our next part on the genetic marker. So in genetic marker, is the differences in the genetic background that can categorize from individual to individual. So that is the definition of genetic marker. For now, we have known many types of genetic marker. We have known about the RFMP. We have now know about the SSLP, AFLP, RAPD, the VNTR. But for time today, we just discuss on the LFRP, which is the restrict fragment length polymorphism, the VNTR and the STR, which is the two types of the microsatellite marker, and the SNP, which is the single nucleotide polymorphism. Wait for me. So first come to the VNTR and the STR polymorphism. So SR, or short time repeat, or sometimes we call it the SSR is sometimes we call it the STR marker, called for short time repeat. So we can see in here the SSR marker is a repetition of different marker. Like in here we have the CTT, 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 CTT. Repeat 1, 2, 3, 4, 5, and 6 times. Okay, and in the another person, another individual, we have this repetition CTT repeat around 8 times. CTT, CTT, CTT repeat 8 times. So when we run electrophoresis, we can see that the movement of genetic marker, because this one have a repetition, so it's a larger size with the repetition of this one. So it will move more slowly during the gel electrophoresis. So and the VNTR, is stand for viral number tandem repeats. So in here, if the STR is stand for short tandem repeat, is a repetition of 2 to 4 nucleotide, and meanwhile the VNTR is a repeat of a region that more than 4 nucleotides. So in here, for example, we have a repetition of a large fragment with 33 base pair. And this marker have 2 locus, locus 1 and locus 2 in the human genomes. So in one person, we have in locus 1, we have 4 repeat of this VNTR marker, and in the other one, we have 6 repeat of the VNTR marker. And in the locus number 2, we have consequently 3 and 7 repetition. And in the second person, we have a number repeat is 2 and 3 and 5 and 4. So if we run electrophoresis, we will have the DNA profile of this VNTR marker. So we have the longest one here, which is the slowest movement, is the 7 repeat, which is longest one. The next one here is 6 repeat, 6 repetition. And in locus here, we have the 4 repeat and 3 repeat. And the DNA profile of this marker in the other person is 5, 4, 3, and 2, consequently. So based on electrophoresis, we will have the DNA profile of this marker in 2 different persons. And based on different profile, we can categorize which one is person A and which one is person B. So for example, in here, we have a marker, which in one person, we have 2 positions in here. For example, in a couple, a parent, we have 6 offspring, okay? We know that in the human nucleus genome, we have discussed in chapter 3 on meiosis and fertilization. Normally, an individual will have a half a set of genome which come from the father, right? And half set of the genome is come from mother. So remember this. So in the mother here, we have 2 locus of 2 marker, one with 3 repetition, and the other allele with 5 repetitions. So the mother in here, we have 3 and 5. So this is the DNA profile of the mother. We have the 5 is the band in the top, and 3 is the band in the bottom. The DNA profile with the fathers here is we have the father have 2 repetition and 1, 2, 3, 4, 5, 6, 7, 8, 8 repetitions. So we have in the father 2 and 8. All right. So 8 was located here and 2 is here. Okay. During electrophoresis. So this couple have 6 offspring. Wow. Congratulations. 6 offspring, and the first offspring will categorize 1 from the father and 1 from the mother. So in this case, C, because this is a girl, because I have a square here, carry the number 5 from the mothers and the number 2 from the fathers. The second daughter carry the number 8 from the father and the number 5 from the mother. The third son is inherited the 8 from the father, and which is here the 3 from the mother. So each offspring will inherit 1 marker from father and the other marker from the mother. Okay. So this one here is the 3 from the mother, and the 2 is from the father, and so on. So come together, we have from VNTR and STR, SSRR or STR are the same. Short tandem repeat or short sequence repeat, the same. So for example, here, if we have 3 marker here, so we will categorize based on this 3 marker, we can have the DNA profile or DNA fingerprint of these 3 marker in different individuals. And this is the example of a real life DNA fingerprint result. We can see in different band here corresponding to each position, each allele of 1 marker. So let's discuss about application of DNA marker, for example, paternity testing. Okay, so here we have a couple, father and mother. We have the DNA for STR marker here. This is the ID of the STR marker. You just go on NCBR or NCBI or ECC, whatever the genome, type the marker here, and it will provide you the position and the sequences of the marker. So in the father, we have two alleles. One is with 11 repetition and one with 14 repetition of the marker here. In mother, we have 8 repetition and 12 repetition. So in child number 1, we will have he or she inherit the 14 from father and 12 from the mother. Okay, the child number 2 is the 8 from the mother and the 14 from the father. And in last child, he or she will inherit the 12 from the mother and the 11 is from the father. So come together, when we combine different loci, we will have different DNA profile of each population. So in 2001, the FBI have deployed the set of 16 STR marker using to identify for paternity testing or criminal investigation. And this is the position and also the ID of each STR marker. We have in chromosome 1, we have none. In chromosome 2, we have 2. In chromosome 3, we have 1. In chromosome 4, we have 1, 2 in chromosome 5, and so on. So I have one question for you. So whether this 16 STR marker can be capable to for paternity testing or criminal investigation in Vietnam population. So this marker have been used by the FBI for forensic science in US population. Now, if we want to apply the same marker to identify the same purpose for paternity testing and criminal investigation in Vietnamese population, it is possible to apply the same 16 STR marker. Anyone? Whether we can use this? You understand the question, right? So I have one answer, yes, we can use the same marker. Do we have another solution? Another answer? So to answer this, we need to go into, let's take an example. Let's take an exercise, okay? So for example, in here, I have a mother and the DNA profile of a child and 3 suspect that, okay? Now, based on, let's say that you are a medical doctor, you are an experiment guy, you are the technical. So with the DNA profile like this, the 3 that, can you have the mother to identify who is the real father of the child? I have first answer is the third one. Anyone? Other solution? Any other solution? So all of you chose the third one. Okay. So to identify this, let's come back to this example. We will have in each child, if the couple is the real father and mother, we will have one marker is inherited from the father and the other one is from the mother, right? So in here, we have the DNA profile of the mother. So if any DNA band is not come from the mother, it must come from the father, right? So this one is come from the mother, right? So the other one here must come from the father. So that one doesn't have this. I'm sorry. Do you have any question? Could you mute yourself, please? I don't know why, but I'm not the host anymore, so I cannot mute you. So could you help me to mute yourself? Bạn Lê Thái Trường Minh có tắt hộ mic được, tắt hộ mình cái mic được đi không nhỉ? Okay, thank you. So we have the band here must come from the father. So that one doesn't have this band, so only that two and that three. So we have this one is from the mother. This one also from the mother. So the other two here must come from the father, right? The real father. So in that number two, he has this one, but he doesn't have the band here. So yes, that number three is a real father here. And now this is the real result. This is a real result from a penalty testing company in Hanoi. So I have taken a photograph of their picture just to take an example for you. I have covered all the name because if I reveal the name of the suspect, I might get the suit due to the law. Anyway, in here, they're using 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 16 marker. And this is the result of the DNA profile of the 16 marker between the father and the son or daughter. I don't know the genders of the daughter. Yeah. So okay. So this is DNA profile of the father and the daughter, and we can see that in the result, the father and the daughter is donor and receive allele in all position of the marker. So there can be denied the relation biological father and daughter with the probability is 99.989%. So the question is why this number is 99.98%. How these numbers was calculated? Do you know how this number was calculated? Can anyone explain to me what is this number mean? Okay. So to calculate this number, we must base on two possibilities. First, inclusion. That means that the child is the real child of the father, and exclusion is negative. The father is not the father of the child. So the paternity index was calculated on each marker by the PI, which the PI is the X, which is the probability that the man transfer the allele to the child. And the Y is the random man out there transfer the allele to the child. So for example, in here, we have the profile of the mother, the child, and the father. So the child inherited the 14 from the mother and 13 from the father. But in here, the father is heterozygous of the 13. So according to here, the father was heterozygous. So in here, the X is 0.5. With this case, with this marker, the first marker in here, okay? And the Y is the probability of a man randomly select, transfer the allele to the child. That means that the ratio of the fathers out there in the society carrying the 13, which is also the allele frequency of the 13 in the society. So in here, Y is 0.308. Okay. So that means the PI in here is X is divided by Y, which is in here the PI of the first marker is 1.62. And the CPI in here is the CPI of all the marker, which is 27,000. And the POP, that means the paternity index, in this case, with calculation by CPI divided by +1, which is 99.996%.
Excuse me, do I need to remember all of this?
No, it's just to explain to you how the number was calculated. That's all. So this part will not in the final exam, if that is your question. Okay. All right. So we come to another tricky occasion, which is the human chimeras. In here, we have a case in which the father with, so we can see in the paper here, a US mom who have together with the wife born a son, but the DNA paternity testing show that he's not the real father of the son. The wife refused that her pay an affair, or in other words, he had a cheating on him. But the DNA test showed that there's a different DNA background between the father and the son. So when the scientists go further on this, they find out that it turned out the father is a chimeras. So the chimeras is the phenomenon which is quite rare, but it appears in human, although it's really rare. So in here, the chimera is fraternal twins. That means the non-identical twin. So in this case, the parent, let's say the grandparent, actually have two non-identical twins. But somehow during the embryonic development, these two was fusion and create one body. Which is also the father in this case. So as a result, the father, half of the father will have a different background, DNA background, and the other half would have a different DNA background due to the fusion of during the embryonic state development. So for example, in here, if the DNA test were taken from one set of DNA and the gamete cell to produce the DNA background of the son will come from the other DNA background, we see that in this case, if we take a paternity test, the result will be negative. That means that the son is not the real son of the father. But actually, it's just a fault dealing to the human DNA chimera. Okay, so we have another application of DNA profiling is the forensic science. So, for example, we have a victim was killed out there, yes, and in the crime scene, they found beside the DNA of the victim, which is the DNA of the killer. And there's 3 suspects who appear at the crime scene at the moment the victim was killed. So in here, we can very easily identify who is the real crime, right? It's the suspect 2, since the DNA here is matched 100% with the DNA in the crime scene. However, the same with the DNA, fraud in paternity testing. Sometimes DNA profile in forensic science can make a wrong conclusion. So you are seeing an example on the wrong conclusion on DNA investigation. So this is Brandon Mayfield, a US lawyer who in 2004, Brandon had a vacation on Spain along with his wife and his son. And in 2004, I remember I was in high school at the moment. There's a terrorist, a bomb was explosion in a train, killed more than 150 people. And in the crime scene, the police had found out the suitcase bomb, and on the suitcase bomb is the DNA of the terrorist. And when they matched the DNA profile firing in the bomb suitcase with the DNA of Brandon Mayfield, it matched 100%. Okay, so as a result, Brandon Mayfield was put in jail because the DNA evidence showing that he is the terrorist. But Brandon have denied he has performed the bombing. Police quite wondering because Brandon seemed to have a beautiful life, beautiful job, and he seemed to have no connection with any terrorist organisms. And he saw no motivation to perform such cold-blood terrorism. But evidence is evidence, and Brandon was put in jail. 6 months later, the police kept the real criminal, the real terrorist who performed the bombing. And when they perform a DNA testing, they find out that the DNA of the real criminal is matched 100% with the DNA sample on the suitcase, and of course, matched 100% with Brandon Mayfield. So Brandon Mayfield have accidentally had the DNA profile matched with the terrorist. So let's come back to our question here. So let's take a DNA background here. Let's say that this fragment have 2 alleles, let's say 15 and 15. So the allele frequency of the 15 out there in society is equal to, let's say, 0.4. That means 40% of the people who out there is carry the allele 15. That means the genotype 1515 is 0.4 square, right? Which is equal to 0.16. And combine each of every one marker, we will have a result like the ratio of the people having this the same profile of this 16 marker is a very small number, which is 0.00001. But imagine that you are living a big city. You are a big city boy, right? So we are living in the city like Hanoi. There's 10 million people out there. So even with the full rate of 0.00001, we have 10 million out there. That means there's around 10 people in this city who having the same DNA background with this 16 STR marker with you. Okay, full conclusion might happen. It is rare, but it might happen. Okay, so remember that in this case, this is a happy ending for a fairy tale story, and it's a happy ending for Brandon Mayfield. But in real, but not every time the story can be have a happy ending like this. Okay, what if the police never catch the real criminal? So remember about this story, because I don't know, maybe it can save you one day. Okay, so we can go quickly on the single nucleotide polymorphism, and then we will have a small break. Okay. So single nucleotide polymorphisms, like its name, is a polymorphism. First, it's a polymorphism. That means it's differences between individual, and it is single nucleotide. That means it involved in only one nucleotide polymorphism. So, for example, in here we have 4 individuals with the DNA sequence like this. So this is one SNP. So the differences between individual 1, 2, 3, and 4 is only one position like this. Okay. And in here we have also another SNP and another SNP. So we have the heterozygous of this one, of the SNP, red SNP for individual 1 and 2, and in individual 3 and 4 we call it homozygous, since the 2 alleles of this SNP are the same between two sister chromosomes. We come to another definition. Is the important definition is the minor allele frequency. So in here, for example, in SNP1, here we have the percentage of allele G. In here is 6. You can see 1, 2, 3, 4, 5, 6. And the number of C is 2, 1, and 2. So the allele frequency of G is 0.75 and C is 0.25. So this is called the major allele, and the C is called the minor allele. And we have a term called the minor allele frequency, which is the second most frequent allele. Remember this because sometimes we have a SNP, single nucleotide polymorphism, with 3 alleles. So in this case, the minor allele frequency will be the second most frequent allele, not the least frequent allele. Okay. Okay, so on the ensembl practice, we have ensembl DNA genome data bank, which contain the information of all SNP that we have been studied. And in the NCBR, we can see that sometimes the allele frequency will be really different between population to population. And that is answer for the question whether we can use the 16 STR marker of the FBI in US to apply in Vietnamese population. Well, due to the differences in the distribution of each marker, the calculation might be not accurate. So to investigate, to apply the same set of STR marker in Vietnamese population, we have to investigate what is the allele frequency of this marker in Vietnamese population and whether it's accurate enough to apply the same marker in this population. Okay. So this is the end of chapter 4. Do you have any question or comment? All right, since you have no more question or comment, let's have a 10-minute break, and then we will come back with the next chapter, chapter 5, okay? Thank you. And easy model to study about inherited laws. So first, let's watch a small clip on demonstration about the experiment of Mendel.
The study of genetics is incomplete without understanding the laws put forth by Mendel. After his experiments of monohybrid and dihybrid crosses, Mendel concluded that these are certain factors which are passed from one generation to the other. But he explained this better with the help of his first law in genetics. The first law in genetics is called the law of segregation. To understand this law, we first need to understand the concept of alleles. We know that genes, in simple terms, are the units of heredity that carry genetic information. Alleles are nothing but the different forms of genes. Let's take a simple example to understand this. Assume we have these two chromosomes. Here we have genes for the character height. Can you tell me which genes could possibly be present here? We can have either both capital TT or both lowercase tt. And we can even have one capital and one lowercase tt as the gene set. Now here, the individual T, that is the single gene, is nothing but the allele. So the alternative form, that is either dominant or recessive, is called allele. In simple words, one single T or T is an allele. Now that we know what alleles are, let's try to understand the law. There are different definitions you will find for this law, but let's pick the simplest one. It says that during gamete formation, the alleles for each gene segregate from each other such that each gamete formed carries only one allele for each gene. It may still sound a bit confusing. To understand this, let's take a simple example. Let's assume we have this single cell with only 2 chromosomes in it. The chromosomes have a set of genes CC for the skin color in an individual. Now to understand the law, let's break it into parts. The first part says, during gamete formation. This is the process of meiosis. It is the type of cell division which gives us 4 cells with half the number of chromosomes. The second part says, the alleles of each gene. So here, C and C is what we are referring to. That means one allele from the gene set. Now what's the next part? The next part of the statement is segregate from each other. This means the alleles will separate. They will not remain together in the process. And now comes the last part. It says that one gamete carries only one allele for each gene. Now this part means that at the end of the meiosis process, we will get 4 cells such that each cell will have only one allele in it. So here, 2 cells will have capital C and the other 2 cells will have the lowercase c allele. Thus, each gamete gets only one allele. To summarize, we can say that the alleles for a gene segregate during gamete formation. To be honest, this law sounds very obvious to us now. It's like having a big book with many chapters in it, and then we make smaller books for each chapter. It is obvious that the smaller books will have the content resembling the content of the big book. This law sounds obvious also because we know the process of meiosis. But nobody knew this back in the earlier times. Hence, it was hypothesized by Mendel and then it was accepted as a law later. So what's the next law? It's the law of independent assortment. Does this mean that the genes assort independently? Or is there anything else that the law specifies? Let's find that out in the next part. There is one common thing among sea waves, dust particles, and a dice rolled on the floor. Any guesses what that common thing could be? It's randomness. Randomness is commonly found in nature and in several things around us. But can you state an example of randomness that occurs inside us? Though our body seems to be organized, there are a few processes that have no fixed pattern. One of the processes is that of the alignment of chromosomes on the metaphase plate. Let me elaborate. Imagine this to be a cell that's about to undergo division. For now, let's assume it's undergoing meiosis. We know that during cell division, a stage named metaphase is reached where the chromosomes align themselves on the equatorial plane. In other words, on the metaphase plate. So can you tell me how these chromosomes will align before they line up on the plate? Will all the red chromosomes from each pair line up on one side, or will they align in an alternate manner? Or maybe like this? Or will the alignment be like this? There are numerous ways in which this can happen. And this is random. There is no fixed pattern or a principle of sequence that the chromosomes follow when they align. Now, if the chromosomes get separated randomly, it's obvious that the genes present on them will also get separated randomly. So let's say this pair has the genes capital A and lowercase a. This pair has the gene set capital B and lowercase b, while this one has capital C and lowercase c on it. We are considering all the sets to be heterozygous in order to understand the concept better. So when the chromosomes get separated in the respective cells, it's obvious that the genes will assort independent of each other. There is no thumb rule that all the dominant alleles will perpetuate in one cell and all the recessive alleles in the other. They can assort independent of each other. This is exactly what Mendel had to say in his second law of genetics. It's named the law of independent assortment. The law says that genes for the different traits assort independent of each other during gamete formation. Let's understand this with the help of this cell. The first part says genes for different traits. This means we are addressing the genes which represent diverse traits. So here we have the gene sets AA, BB and CC on these respective chromosomes. These are the genes for different traits. Now the second part states assort independent of each other. This means the genes will not have a fixed pattern to follow when they segregate in the cells. So here, the alleles of the gene set AA will assort independent of the other two gene sets. It's not mandatory that all the dominant alleles will get assorted in the cells together. So the allele capital A can get assorted with any allele from the gene set BB and CC. And that's the reason why we get the gametes with different possible combinations at the end. To quickly review, the assortment of alleles is purely random and does not have a predictable and fixed pattern during the process of gamete formation. That is what the law says. There can be more possibilities of obtaining different combinations depending upon how the chromosomes align themselves on the metaphase plate. Now tell me, which type of a cross will help us understand this law better? A monohybrid cross or a dihybrid cross? Think about it. Let me help you with this. If it's a monohybrid cross, then there will be no independent assortment of the different alleles. And why is that so? That's because we are considering only one character at a time. So we will be dealing with a single gene set. And thus, this law is best understood when there are more than one character studied at a time. This is nothing but the dihybrid cross or higher crosses which involve more characters. Now that we're well versed with the first two laws of genetics, let's have a look at the third law in our upcoming part. A scene of lush green trees covered with yellow or brown mesh is not new to us. We have come across it many times. Any idea what this mesh is all about? These are also plants, parasitic plants to be precise. Being parasitic, they derive all the nutrients from the host and grow to form a huge structure. These plants are like creepers and overpower the huge trees as they grow. This is one of the best examples for understanding the fact that dominance is not a matter of size. Rather, it's never affected by size. It's the nature of the dominating component that matters. Let's take our genes for example. We know how small genes and their constituent alleles are. Yet, there is one form which dominates the other. We are familiar with the concept of dominant genes and recessive genes. But this wasn't known back then, not even to Gregor Mendel. That is the reason why he put forth the concept of dominant and recessive alleles with the help of this law. This law is called the law of dominance. After the monohybrid cross, Mendel concluded that a few genes are dominant while the others are recessive. Let's have a look at what the law has to say. The third law of genetics, known as the law of dominance, states that some alleles are dominant while others are recessive. An organism with at least one dominant allele displays the effect irrespective of the presence of the recessive one. The law is actually very simple. Let's take an example and understand it. Let's consider a cross of a tall and a dwarf plant. The first part of the law says that some alleles are dominant while others are recessive. Here, can you tell me why is this plant tall? That's because it has both the dominant alleles. We represent it with the capital letters TT. Similarly, this is a dwarf plant as it has both the recessive alleles denoted by TT in the lowercase. Now the next part of the law states that an organism with at least one dominant allele displays the effect irrespective of the presence of the recessive one. So what does that mean? Here, on crossing the two, we get these plants in the F1 generation. The phenotype of all these plants is tall. And what is their genotype? As we can see, they are all heterozygous and with one dominant and one recessive allele. Doesn't the tall phenotype clearly indicate that the dominant allele has masked the presence of the recessive one? That's right. That's what the second part of a law says. The presence of a single dominant allele is enough to express the trait phenotypically. And what if both the genes are dominant? It's obvious that the phenotype will be dominant. The law states at least one dominant allele, which means If one or even both the alleles are dominant, then we get the respective dominant phenotype. Now let's cross this F1 generation to check whether we have understood the law correctly. The cross of these F1 hybrids gives us these offsprings in the F2 generation. Can you help me with the phenotype and the genotype of these offsprings? Here we get 3 tall plants and 1 dwarf plant. Genotypically, there is one plant that is homozygous for the dominant trait. There are 2 plants which are heterozygous, and this last one is again homozygous but for the recessive trait. So can we relate these results with our law of dominance? There are 2 types of alleles, one dominant and one recessive. This is what the first part says. And the second part says that at least one dominant allele displays the effect irrespective of the presence of the recessive one. So as we can see here that these 3 plants are tall because they contain the allele T for tall trait. This one has both the dominant alleles, and these 2 have one allele for the dominant trait. Its presence masks or suppresses the effect of the recessive allele. So this was about the third law of genetics, called the law of dominance.
Okay, so in the clip they have divided a little bit different with me. They have the first is the law of segregation, the second is the law of independent assortment, and the third is the law of dominance. I just turn the law of dominance come first. Okay, it's not a problem. So in Mendelian, he have choose to study the pea plant. So one fact about Mendelian, about Mendel, is the family of the parent of Mendel is really poor, and they are so poor that cannot afford the expensive education at the meantime. So Mendel was sent to the church to become a priest, so that his education would be paid by the church. So he don't have to worry about the money, but in return he will not have having the family. But because he didn't allow to have family, he didn't have any offspring. So he did not have to worry about them. So he has time for science. Anyway, he has used a pea plant as a model. When he's performed his experiment between 2 pea plant, one have the purple flower and one has a white flower. The parent is true breeding parent. That means that they are homozygous with the allele corresponding to the color of the flower. So in F1, he have obtained all the plant have the purple color. So he had invented the first law, which is the law of dominance. So which he have some term that if organism or plant have 2 identical alleles or genes, it's called the homozygous state. And if the 2 alleles were different, it's called the heterozygous state. And in here, because the parent is true breeding, so that means they are both homozygous. So in F1, we have 100% of the white one is heterozygous with 1 dominant allele and 1 recessive allele. And the allele that corresponding to the phenotype show in F1 is called the dominant allele, since it's dominant the other trait causing by the other one. So he has studied in 7 traits of the pea plant: the flower color, the purpose white, the position of the flower, the seed color, the seed shape, the pod shape, pod color, and the length of the plant. And he have take the conclusion that in each pair, there will be one phenotype is considered as a dominant phenotype, and the other one is the recessive one. So, for example, the purple color is totally dominant against the white flower color, or the yellow in the seed color of the pea plant is dominant against the green. The shape round is over dominant against the shape wrinkle in the pea plant, and so on. So the second law of Mendel is the law of segregation. So when he's continued to take a cross, for example, in here, he take a trait pot shape, green versus yellow, both is true breeding. So F1 is 100% have a green pot, like the first rule, the law of dominance. But when he take F1 or cross-pollination, he take the F2 with the ratio 3:1. 3:1. So 3 has a green pot and 1 is has a yellow pot. So in here, he invented law of segregation. That means that in here, when the F1 was self-pollination, it's getting to the combination between X gamete. The big G here is corresponding for the green pot, and the small g is according for the yellow pot. And as a result, we will have the genetic ratio is 1 big G, 2 big G, small g, and 1 small g. And with the first two genotype, we have 3 big G plus here. That means here is the green part, and 1 here is GG have the yellow part. Okay, and this 3:1 ratio is continued when he's study in all of his 7 traits in the pea plant. So one fun fact about Mendel. So when he analyzed the phenotype trait of the pea plant, he hired a gardener. Like right now, I have a research assistant, something like that. And until now, we don't know if he did it in purpose or not, but he told the gardener what is the expected number of X phenotype in the offspring. And in the effort to keeping his job, the gardener tried to make the number so beautifully, so perfectly, by throwing away all the offspring that not following the 3:1 ratio. So the number is so perfect until 200 years later, a statistician, Fisher, have imagined that why those number is so perfectly, and he start to calculation what is the probability of obtain such so perfect number like this. Even the theory of Mendel was right, and he goes to the conclusion that even with the right theory, it is impossible to obtain. It's nearly impossible to obtain such a perfect number like this. So Mendel, father of genetics, were catching cheating in science by Fisher, who considered as the father of statistics. All right. So Mendel is the first case of being cheating in discovered to be cheating inside. Remember this. Anyway, the 3-to-1 ratio was replication in all the traits of the pea plant, and until now, scientists was agreed that Mendel actually very lucky because this trait was located in the different chromosome. So maybe if he choosing the other trait, if those trait is follow in the same chromosome, then it's not following the 3-to-1 ratio. So it might confuse him, but maybe he chose this phenotype on a purpose, not randomly. Anyway, when he performed dihybrid code, that means he chose not only one phenotype, but 2 different phenotypes in his cross. And he find out that if we follow the same rule, in F1, we still have 100% of the offspring showing the dominant phenotype. But in F2 generation, we start to have the variation of the 3-to-1 ratio, but now powered by n, with n is the number of the phenotype. So in here, we choose 2 phenotypes, the color of the pea plant and the shape of the pea plant. So number in here is 2, which is 3-to-1. So power by 2 is 9-to-3 to 3-to-1, which 9 have the ratio of 9 having a yellow and round pea plant, and 3 have each of a recombinant phenotype of the first parent here, and 1 is showing the recessive phenotype. Okay, so this is the table of contribution, which you have already done it in high school. I will not repeat it. So in the third law of separation of Mendelian, he's find out that in pea plant, in also other species, each pair of allele was separated independently during meiosis and combined also randomly in fertilization. And it leads to the process that all the F2 generation will follow the 3-to-1 power by n, and n in here is the number of phenotype that we put in the research. So this small exercise, but we don't have the time for exercise, so you can take a screenshot of the exercise and try to handle it at home. Okay. So the summary of Mendel rule. So we have the rule of dominant, which is in F1, if it's the true breeding parent, then in F1 we have 100% will show the dominant phenotype. The law of separation, if we continue to make a F1 autoplanary in the F2, we will have the ratio 3-to-1. 3 is showing the dominant phenotype and 1 is showing the recessive phenotype. And independent assortment, which is in the different phenotype, will follow contributing to the 3-to-1 ratio. But concerning the number of phenotype will be follow the distribution of 3-to-1 power by n, with the n is number of phenotype. So we come to the new part, exception for Mendelian rule. So the first exception of Mendelian rule is the mitochondrial DNA. So we discussed about mitochondrial DNA. We know that it's inherited follow the maternal pathway. That means that in the nuclear DNA, so each offspring will inherited a half of chromosome set come from the mother, and the half of chromosome Z, it comes from father. But meanwhile, in the mtDNA, we only inherited almost, let's say, more than 95% of the mitochondrial DNA comes from our mother. And the mother is inherited from her mother and the mother before her. So it's a maternal lineage.
Humans inherit mitochondrial DNA only from the mother. So, in order to pass on mtDNA from generation to generation, the mother must have an unbroken link of female descendants. Unlike nuclear DNA, which undergoes recombination, much of mtDNA is passed from mother to daughter, relatively unchanged. If we take a sample of mtDNA from a population and try to reconstruct their matrilineal ancestry using the very slow mutation rate of mtDNA, we might arrive at a single female common ancestor, a mother whom we can all relate to from our mother's side. This is exactly what Can, Stoneking, and Wilson did in 1987 and found out that, in fact, all humans alive today do have a common female ancestor who probably lived about 200,000 years ago in Africa. They named her Mitochondrial Eve. Analogous to this, tracing Y chromosomal DNA, which is only inherited from father to son, points to our common father, Y chromosomal Adam. However, there is no reason to believe that mitochondrial Eve and Y chromosomal Adam lived at the same time. Likewise, they were not the only humans living at the time, but their contemporaries failed to produce a direct matrilineal or patrilineal descent. Also, mitochondrial Eve and Y chromosomal Adam were only the most recent common ancestors. Going back in time, we can even find common ancestors of species all the way back to the first single-celled organisms. What's more, our actual common ancestor following both mother's and father's side probably lived as early as 5,000 to 10,000 years ago. All this shows that we are so closely related, more so than we could ever imagine.
So in the term, we are maybe more close than imagined. We will have a very funny discourse about this later. So recently, a paper published in 2016 to investigate why all the mtDNA was inherited from our mother. Of course, because in the age, almost of the mitochondria is inherited from the age, and the age it comes from the female gamete. But in sperm, we have mitochondria too. So why these mitochondria from the sperm cannot be inherited to the offspring? So a research published in 2016 showing that in a frog model, so that when the sperm inject into an egg and start to inject the inherited material inside an egg, it have activate a protein called the CSP6 gene, which will cleavage all the mitochondria of the sperm. So as a result, all the mitochondria from the sperm will be destroyed when the sperm have a combination with an egg. So let's have a small clip on demonstration on the disease that we inherited from the error in mitochondrial DNA.
Mitochondrial diseases cover a wide range of conditions caused by genetic mutations. In the UK alone, they affect thousands. Mitochondria are the powerhouses of the cell. They use a process called oxidative phosphorylation to generate energy. But a mitochondrion is a complex piece of machinery, and a lot can go wrong. It takes around 1,500 proteins for mitochondria to function, and problems with many of these can lead to disease. The cell's nuclear DNA creates all but 13 of these proteins. The rest are coded for by the DNA in the mitochondria themselves. And mutations in either can cause problems. A cell has just one nucleus and many mitochondria. All a cell's mitochondria contain their own DNA. If a mutation affects mitochondrial DNA, some might be healthy, others defective. And different cells can have different numbers of defective mitochondria inside them. That means that some cells may be less affected than others. The diseases caused by damaged mitochondria can also be varied. They can affect any organ or tissue, and they can appear at different times throughout life. For example, Leigh syndrome is the most common type to develop in childhood. It can affect patients in a number of ways, including loss of skills, epilepsy, and problems with muscle function. Mutations in over 75 genes in both nuclear and mitochondrial DNA can lead to this syndrome. So what can be done to tackle mitochondrial diseases, like Leigh syndrome? Treating patients is difficult, but in some cases, the disease can be stopped before it's passed on. Mitochondria are inherited from the mother. If the mutation is in the mitochondrial DNA, this inheritance can be blocked. One approach is to combine the nuclear DNA from an affected egg cell with healthy mitochondria from another woman. The embryo would then have nuclear DNA from the original mother and father, and mitochondrial DNA from the egg cell donor. This has led some to call such potential infants three-parent babies. This technique is still very new, but it has recently been approved in the UK. It may be a controversial approach, but techniques like these could finally help combat these devastating diseases.
So the technique they has mentioned in the clip, the three-parent technique, actually have been performed to treat the disease Leigh syndrome. So Leigh syndrome is a disease that causing mostly by the mutation in the mitochondrial genome. So in case here, a mother with Leigh syndrome and defective mitochondria have been follow the three-parent technique here to create a healthy boy, in which the doctor first will so in here the paper say that the mother have two children already dead because of the Leigh syndrome. So in the third one, he decided to intervene, decide to treat with the three-parent technique, by which so this is the mother. So this is the DNA of the mother, and he has a normal nucleus genome. But the defective mitochondria. Okay. Also, he has some normal mitochondria. So this is the empty carry later. So to treat it with this, first the scion will take the nucleus out. Nucleus of the mother taken out. The normal nucleus genome and put it into another mother or another healthy woman who have normal genome who have normal empty DNA genome. So the nucleus genome of this woman was taken out, and the nucleus genome of the mother was taken in replace of here. We call it the egg donor. Okay. Egg donor. Was replaced by the egg, by the nucleus of the mother. So now we will have the nucleus here of the mother and the mitochondria here of the healthy woman. And it will combine with the sperm of the father. So this is the sperm from the father. So this is why this is called the three-parent technique, because the new baby actually have three parents. So the nucleus here is come from mother. The mitochondria here is from the another woman, and the sperm provide also another haploid of nucleus genome. So this baby actually have the genetic material from three different individuals. So that's why we call it three parent. Okay.
So is this similar to cloning?
No, it's not similar to cloning. Cloning is like the dolly ship, right? Like you want to mentioned about cloning, like the cloning in the Dolly strip, or the cloning in the create recombinant genes. So, for example, in the the cloning of the Dolly strip, I believe that this is what you mean. So all the genetic material is come from one parent, but they just erase all the cell memory, all the cell memory of the egg to create the new cell line, and it's putting it just borrow the chamber, like the womb of the other parent, just borrow the womb of the other parent. The Dolly strip doesn't have any material from the second one, and in here it had the genetic material from 3 different persons. Okay, the nucleus is from the real mother and the real father, and the mitochondria from another mother. All right. So I have received a question. Please, when you send a question for me, please send it us to everyone, okay? So I will copy the question here. So if anyone interested. But we have many egg cells. What if the sperm cell attacks the wrong one again? All right. So in scientists, if we take it in silico, we call it in situ fertilization. So the egg will be taken out of the body of the mother, and then we handle it. We make sure that only eggs were taken out, only one egg cell was taken out of her body, and then we extract the nucleus genome and put it into the nucleus with the eggs from the healthy donor, which we have destroyed the nucleus of the cell. So they kind of make sure that there's no other age inside. Cái này cũng giống như là ở các bệnh viện bây giờ khi mà người ta làm cái thụ tinh nhân tạo đối với các cặp vợ chồng mà hiếm muộn ấy, hay là vô sinh hay hiếm muộn ấy, đúng không? Thì đầu tiên là họ sẽ tinh trùng thì dễ rồi, đúng không? Tinh trùng thì cho mỗi ông một cái cốc vào một góc nào đấy là ra được một đống. Nhưng mà trứng thì khó hơn. Trứng thì mỗi một chu kỳ thì người phụ nữ 1 tháng chỉ dụng 1 quả thôi. Dĩ nhiên là cũng có trường hợp dụng 2-3 quả, giống như là sinh đôi khác trứng này thì thỉnh thoảng có dụng 2-3 quả, nhưng ít. Thông thường là một chu kỳ là 1 tháng là chỉ quả trứng thôi. Thế thì để mà tăng cái xác suất mà thụ thai ấy, thì không bao giờ người ta cấy một phôi vào cả, mà người ta sẽ cấy vài phôi. Và để cấy vài phôi ấy, thì người ta sẽ tiêm hormone, tiêm estrogen vào cơ thể người phụ nữ. Nhiên là có hại, nhưng mà thôi, có con thì phải chấp nhận thôi. Thì khi tiêm vào thì người phụ nữ sẽ trong một chu kỳ thì dụng đến vài ba quả trứng, có trường hợp 4-5 quả gì đấy. Thì sau đó người ta sẽ lấy các quả trứng này thụ tinh với cả tinh trùng của ông chồng, và sau đó thì để phôi phát triển. Những cái chương trước thì mình đã dạy các bạn rồi, đúng không? Tế bào gốc đầu tiên sau khi tinh trùng kết hợp với trứng thì sẽ chu kỳ phân bào nó sẽ là 24 giờ, nghĩa là 1 ngày. Sau 1 ngày thì nó phân đôi. Thì những cái phôi đấy ấy, thì cứ sau 1 ngày nó phân đôi, 2 ngày thì nó phân thành 4 tế bào, 3 ngày nó phân thành 8 tế bào. Thì người ta sẽ đợi đến khi phôi được ngày 5, có nghĩa là khi mà đã phân bào thành 2 mũ 5 là tương đương với giai đoạn là 32 tế bào. Thì người ta bắt đầu đi tách một cái tế bào ra để người ta đi giám định gen, xem là những cái phôi đấy có bị cái bệnh tật gì không. Ví dụ như cặp vợ chồng bị một cái bệnh hiểm nghèo nào đấy mà muốn loại những cái phôi lỗi đấy ra khỏi cái gia đình của mình, thì sau khi người ta giám định gen xong, người ta thấy là sau khi tổ hợp xác suất các cái version của gen, bởi vì là học Mendel thì biết rồi, đúng không? Phân đi độc lập, tổ hợp tự do. Nếu mà có một cái gen lỗi thì có 50% là xác suất là các offspring sẽ thừa kế 50 cái gen lỗi đấy, còn 50% còn lại là không. Thì người ta sẽ xem là trong những cái phôi còn lại, phôi nào phôi khỏe thì người ta cấy vào cái phôi khỏe vào tử cung người mẹ thôi. Bà mẹ thì cứ nằm ở đấy đến khi sau khi cấy vào thì Dĩ nhiên là phôi cấy vào thì nó sẽ không được khỏe như là phôi mình thụ tinh theo phong cách bình thường. Thì bà mẹ sẽ phải nằm kiểu treo chân dưỡng thai trong vòng mấy tháng đấy. Sau khi nó đậu vào tử cung và bám chắc vào cái niêm mạc tử cung, niêm mạc tạo thành tổ và bám chắc vào cái niêm mạc tử cung rồi thì mới có thể sinh hoạt bình thường. Thì sau đấy thì quá trình sinh nở thì như bình thường thôi. Okay, so let's come back to our So another exception of Mendelian rule is the incomplete dominance, where in the F1 generation, although carry the heterozygous genotype, it doesn't show the phenotype of the dominant, but show the intermediate phenotype, which is the, let's say, a median phenotype between the dominant and the recessive. So in the incomplete dominant, we will have the ratio, instead of 3:1, will be 1:2:1, exactly like the ratio of the genotype. And codominance, for example, if a gene have more than one allele, like the blood type in human, we have the O is recessive, the A and the B is a codominance. So we have two alleles as dominant here, and the AB is the heterozygous between IA and IB. So in the blood type, so in the membrane of the erythrocyte will be receptor, and in the plasma then will be antibody. So in blood type O, we will have plasma against the A and B. So in blood type A, in the membrane, we have the A antigens, and the antibody is against B. In the B, we will have the B antigen, and the antibody is against A. And in AB, we have none antibody in the plasma, and in the erythrocyte, we will have both A and B antigen. So if you are watching the old movie, bạn nào ngày xưa mà đã xem cái phim là Bao Thanh Thiên ấy, cái phim đấy thì chiếu từ thời rất là khi tôi còn bé này, thì không biết đến thời các bạn thì có xem nữa không. Tôi cũng không biết là Bao Thanh Thiên thì dịch sang tiếng Anh là gì. Thì có một cái tiết mục rất hay là nhỏ máu nhận thân, đúng không? Là một ông nào đấy nghi ngờ thằng cu này không phải con mình, là cắt một giọt tiết ra, thằng bố cũng cắt một giọt tiết ra cho bát nước xem là hai giọt máu đấy nó có hòa lẫn với nhau không. Nếu hòa lẫn với nhau mà không vón cục thì là khẳng định là bố con, đúng không? Thì mà nếu mà nó vón cục với nhau hay là tạo thành cái hiện tượng gọi là đông máu ấy, thì là không phải bố con. Thế thì cái tính khoa học của cái hiện tượng này là đến đâu? Thì ở đây các bạn biết là trong máu thì nó có hai phần, một là kháng nguyên trên bề mặt hồng cầu như mình vừa giải thích, và một hai là cái kháng thể trong cái plasma, có nghĩa là plasma huyết tương. Thế thì ở trong huyết tương của nhóm máu A thì có huyết tương có kháng thể là kháng B, và trên bề mặt hồng cầu là kháng nguyên A, đúng không? Thế thì nếu mà A mà gặp B thì sẽ cái kháng thể này kháng gặp phải cái nhóm máu A thì nó sẽ bón cục lại, tạo thành hiện tượng đông máu. Thì ở đây nếu mà bố với con mà có cùng nhóm máu, chẳng hạn bố mà đều nhóm máu O, con đều nhóm máu O, thì đương nhiên là máu nó tan. Vậy trường hợp này là về mặt khoa học, về mặt chẩn đoán theo kiểu ngày xưa, lẫn về mặt khoa học là đúng là bố con thì rõ rồi. Nhưng mà trong trường hợp là ví dụ như là bố là nhóm máu O, nhưng mà mẹ lại nhóm máu A, sinh ra con là nhóm máu A chẳng hạn. Đấy. Thế thì khi nhóm máu bố mà nhỏ vào nhóm máu con, trong trường hợp đấy thì là bởi vì trong kháng nguyên của nhóm máu O là có kháng thể kháng A, mà trong máu của con lại là có kháng nguyên A, đúng không? Như vậy là sẽ tạo thành hiện tượng đông máu. Như vậy là về mặt logic, về mặt sinh học thì đây vẫn là bố con bình thường, đúng không? Thời đấy thì chưa có giám định gen hay là 16 chỉ thị SDA gì cả, mà chỉ có mỗi nhỏ máu thôi. Thì là trong trường hợp này thì vẫn là bố con, nhưng mà nếu mà nhận máu theo cái phương pháp ngày xưa đấy là nhỏ máu nhận thân đấy thì máu vón cục, như vậy kết luận là đây không phải bố con, đấy là kết luận sai, đúng không? Okay, so another exception of the Mendelian rule is the polygenic trait, which is a trait was corresponding to many different genes in the genomes. So in this case, it also not following the rule of Mendelian, since in Mendelian, one trait is corresponding by one gene or one loci. Another exception is epitasis. One phenotype was corresponding by two different locus, but different with the polygenic. In here, one pair of the genome, one pair of the allele will dominate against the other one. So for example, in here, we have, if the locus one in position is EE, it will dominate and prevent the second allele, the second locus to show the phenotype. So thanks that, and as a result, both individual here will have the yellow fur. And in case of the big E here, so there is no epitasis phenomenon, and there is no prevention of the second locus showing to phenotype. So if the individual have a small b, it will have a brown fur, and a black b, big B, it will have a black fur. So in case here, the black is dominant against the brown without any epitasis from the first locus, the E here. Okay, so in human disorder, we have to get familiar with the symbol of human familiar symbol. So you may know that in high school, male, female, here is the marriage here, and we have in here some case like close marriage. So just a small question. You all know about close marriage, close relationship. Sometimes, in some cases, it's called incest or Alabama special relationship, whatever. Anyway, in your opinion, is close marriage or related marriage is have any effect on? Of course, it have effect on the offspring, but it's a good thing or it's a bad thing? Yes.
Yeah, objectively depends. Like it's bad for someone, but yeah, it's usually bad for one.
Okay, so wait for me a minute.
Oh, unless
Yes, any opinion? So we all know that incest is a bad thing. The offspring of the incest couple will have more chain to show the recessive phenotype, and not only recessive phenotype is bad, blah, 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 and so on. The vitality of the offspring, which is result of the incest relationship, is less vitality compared to the normal one. Okay, all of us agree that it has been demonstrated in not only on human model, but also in plant model and also in animal model. Okay, but let's go back on some mathematical. Okay, so to born you, for example, to born an individual, here we need two individuals, right? Father and mother, right? So to produce one individual here, we need two unrelated individual, right? And to create two unrelated individuals, so our father have his own father and mother. Our mother has also his own father and mother. So on. So to create one individual nowadays, in one generation we need two unrelated individuals. 200 generation we need 4 unrelated individuals. And in n generation, if we want to make sure that there is no any close marriage, any incest in our family, all the individuals in the family are unrelated, we will have to have 2 powered by n unrelated individual, right? And let's suppose that n is equal to 64. That means in 64 generation, if we don't want any unrelated individual in our family, we will need 18 times 10 power 18. That means 18 billions people to create one individual. So 64 generation, let's suppose that one generation is equal to 25 years. So it must calculate from the time one was born and until he or she can get married and make an offspring. So 64 generation equal to how many years? Please, 16,000 years. Sorry, 1,600 years. Okay. And 18 billion people is much more than all the people have been existing on Earth. Okay. It's more than all the people So, as according to the scientists, the people have appeared on Earth like the human was appeared on Earth like 10 million years ago, and all the human have appeared on Earth who have count also the people have been existing nowadays, and the people have been dead is around 107 billion people have been appear on Earth since the day humans have appeared on Earth. Okay, so in 1,600 years, we need the people much more than all the people have appeared on Earth 10 million years ago. Come on, we don't need to be a rocket scientist to know this is impossible. So it turned out that there is many incest relationship in the human history, and the scientists have calculated and they have provided truth that if you are all your families was belong to one nationality, that means that if you are a Vietnamese, your parent is Vietnamese, your grandparents are Vietnamese, all of your family members are Vietnamese, there will be a high chance that you will have in your family tree will have an incest relationship within 6 or 7 generations. They sure about that. Okay. So sum up, incest is bad. We know it. It has been proved in animal and human model. But thanks to incest, human was existing today. Okay. Bạn có thể biết là trong lịch sử của Việt Nam ấy thì cái triều đại mà có thể gọi là có nhiều cái mối quan hệ kiểu giao phối gần như này nhất chính là triều đại nhà Trần, đúng không? Triều đại nhà Trần thì lại chưa đến nỗi mà anh em ruột lấy nhau đâu, nhưng mà gọi là anh em con chú con bác, gọi là họ hàng lấy nhau thì nhiều vô số kể. Thực ra thì nếu mà mình tính ngược lại cái bài toán ấy, cách đây ví dụ 1.600 năm thì tương đương khi mà nhà vua Lý đầu tiên ấy, Tiền Lý rời đô từ vua Lý lập ra nhà nước Việt Nam, thời đấy là nước Giao Chỉ, thì dân số Việt Nam lúc đấy chỉ đâu đó khoảng độ 1 triệu người, đúng không? Chủ yếu sinh sống trong các làng xã và theo quần thể. Thì các bạn nào mà về quê thì biết rồi, ở quê thì hầu hết là đầu làng cuối xóm là hầu như là có họ hàng với nhau hết, cho nên là thanh niên lớn lên thì xung quanh là toàn là họ hàng cả, thì chắc là cũng không có nhiều sự lựa chọn. Các cụ nhà mình chắc cũng không có nhiều sự lựa chọn. Okay, so I guess that today this is the end of today's session. Do you have any question or any comment?
No question, no.
Okay. Okay, so since you have no more question or comment, thank you very much for your attendance. Thank you for have come along with me in, I don't know, 3.5 hours. And thank you for your attendance, and see you next week. And in next week, I will spend around, I don't know, half an hour or 1 hour to have a quick recap, summary. What is the most important part that we have been discussed from the beginning, chapter 1 to now? Okay, so pay attention. Thank you very much, and see you next week.
Goodbye, teacher.
Well, Professor.
Bye, Professor.
Bye, Professor.
Yeah, do you have any question? You can turn on your